Okay, in this video we're going to be examining the process in chemistry of cholesterol synthesis. So let's start by taking a look at where the substrates for cholesterol synthesis come from. Now the major organ that is going to be producing cholesterol, of course, is the liver. All cells can produce cholesterol, but the liver is the major source of cholesterol in the body for biosynthesis. Now, where do the substrates come from for cholesterol synthesis? The pathway is going to be very similar to fatty acid synthesis. So because we are generating a very large molecule of cholesterol, we are going to be doing this in an anabolic state. So this means that we will be in a fed state with insulin signaling. Once again, the same principle holds true as from synthesizing fatty acids and proteins, is that we want to do this in an anabolic state because synthesis of cholesterol takes a lot of reagents and a lot of energy. So we want to do this when we know that we have plenty of fuel sources coming in the fed state. So the original source of the material for cholesterol is going to come from glucose. So the glucose concentration in the fed state is relatively high. This will enter the liver cell through the GLUT2 transporter, and it will be processed by glycolysis. Glycolysis, the endpoint, of course, is pyruvate. The pyruvate now translocates into the mitochondria, and in the mitochondria, the enzyme pyruvate dehydrogenase, of course, converts this into acetyl-CoA. So the process so far is very similar to what we've seen to fatty acid synthesis. Glucose is going to be our original source of carbon molecules. Now, once we have acetyl-CoA, this will, of course, condense with oxaloacetate into the TCA cycle, and we are going to form citrate. Now the citrate, like in fatty acid synthesis, is going to be exported into the cytoplasm. And this, of course, is going to be done by the citrate malate transporter or shuttle system. So we have high energies, high NADH concentration in the mitochondria, in a fed state, anabolic conditions in the liver, and therefore the isocitrate dehydrogenase enzyme will be inhibited, as we saw in fatty acid synthesis, which will increase our concentration of citrate. The citrate is then transported into the cytoplasm. Now in the cytoplasm, our citrate lyase enzyme will process the citrate into oxaloacetate and acetyl-CoA. Now, once we have our acetyl-CoA in the cytoplasm, this is where we start forming either fatty acids, or we can use the acetyl-CoA to generate cholesterol. So recall, for fatty acid synthesis, the acetyl-CoA molecule needs to be converted into malonyl-CoA. Then the acetyl-CoA and malonyl-CoA are processed by the fatty acid synthase complex to generate ultimately our C16 palmitate fatty acid. Now for cholesterol synthesis, we're going to take a slightly different path for our acetyl-CoA in the cytoplasm. So let's take a little closer look at these reactions. So the first step in cholesterol synthesis is we are going to be taking two acetyl-CoAs, and we are going to be condensing these two molecules together by the enzyme thiolase. This thiolase, recall, we have seen in fatty acid synthesis. This is the final step to make a fatty acyl-CoA molecule in fatty acid synthesis. We will be using the coenzyme A to displace the newly synthesized fatty acid off of the fatty acid synthase complex. So this is simply a reversal of the reaction, a slightly different take on the reaction, and what we will be generating is this molecule. This molecule is called acetoacetyl-CoA. And again, it's simply condensation of two acetyl-CoA groups into this molecule. Now the next step of this reaction is that we are going to be adding another acetyl-CoA to have a final count of 3 acetyl-CoAs in one molecule. We're going to be adding this acetyl-CoA onto this carbonyl of acetoacetyl-CoA. And in doing so, we will generate this molecule. Now notice here that the hydrolysis of this thiol ester bond of the coenzyme A is driving the reaction. So in this reaction we are also losing a CoAsh from the acetyl-CoA. The enzyme that performs this reaction is called HMG-CoA synthase. And what we are forming is HMG-CoA. What HMG stands for is beta-hydroxy, beta-methyl, gluteryl CoA. Now where does this name come from? What we have is an example of naming our carbons as we have done with fatty acids. This particular carbon is the beta carbon. So recall the beta carbon is number 3 past the number 1 carbon being the carboxylic acid. So 1, 2, 3, alpha, beta. So what we have here is beta-hydroxy, our hydroxyl group on the beta carbon, beta-methyl, methyl group on the beta carbon, gluteryl. And the gluteryl structure, if you recall the structure of glutamate, off of the carbon here we have a carboxylic acids on both ends, separated by 2 carbons. So this resembles the amino acid glutamate. Now it's important to note that up to this point, all of these reactions are fully reversible. And this has a very important consequence when we talk about regulation of cholesterol synthesis and drug intervention for controlling cholesterol synthesis. So the reaction by the thiolase and HMG-CoA synthase are both reversible reactions to generate HMG-CoA. Now it's at this point, the next step in the reaction, that is irreversible, and we are going to be forming the main component of the molecules we need for cholesterol synthesis. What we are going to be doing now is performing a redox reaction. And in this redox reaction, we are going to be reducing one particular portion of this molecule, and that is this carbon. In fact, we are going to be performing two reductive steps. So ultimately, what we are going to be having here is this structure. Now notice what we've done for this carbon. We've added two hydrogens and converted the ketone structure, the carbonyl, into an alcohol. Now in order to do this, the electrons between the sulfur group and the carbon of the carbonyl, these electrons will be held by the sulfur group of coenzyme A. What this will generate is our CoAsh, which will be a leaving group. In doing that, that leaves our carbon deficient in a total of 4 electrons. To fill this deficiency, what we are going to do is use the electrons from 2 NADPH. So in 2 successive reduction reactions, we are going to be adding hydrogen with 2 electrons to this carbon to give us our final structure of our CH2OH. Now the enzyme that performs this reaction is called HMG-CoA reductase. And what we are forming here is the molecule mevalonate. Now, the reason this step is very important in cholesterol synthesis is because this step is not reversible. Of course, it's not reversible because we are using 2 reducing equivalents of NADPH in order to generate mevalonate. The free energy change is far too negative to reverse this. So this turns out to be a very important step for medical intervention. If we want to control cholesterol synthesis, reduce cholesterol synthesis in the liver, it makes logical sense to be able to regulate this enzyme by drug intervention. Because this is the irreversible step which commits us into cholesterol synthesis. If we block this step by inhibiting HMG-CoA reductase, we can build up the concentration of HMG-CoA, and then the 2 previous reactions are reversible. If these are reversible, that means that ultimately with this reversal, these 2 acetyl-CoAs, along with the third one, can be put into fatty acid synthesis. So essentially, we will not be building up any toxic intermediates, but we have a way to get rid of our intermediates by shunting them into another pathway. So what is a good strategy of inhibiting this enzyme? These are the statins. So these statins are used in individuals with high cholesterol levels in order to reduce their endogenous cholesterol synthesis in their liver. So the statins, taken once a day, inhibit this enzyme and inhibit the conversion of HMG-CoA into mevalonate. Okay, now let's take the next step. Now that we've committed ourselves into cholesterol synthesis, what happens to mevalonate? This is where cholesterol synthesis begins to get very complicated. So what I want to do is show you just a few important intermediates in this next few steps to give you an idea of the complexity of cholesterol synthesis. So let's take a look at this product. So this product here, this is called isoprene pyrophosphate. Isoprene pyrophosphate, notice, is a 5-carbon molecule. So we have here in mevalonate 1, 2, 3, 4, 5, 6. So clearly, in order to generate isoprene pyrophosphate from mevalonate, this is going to be several reaction steps. These reactions will include oxidative reactions. Notice the double bond that's introduced. We are going to lose a carbon, so there's going to be a CO2 right here released. And so there are several steps to convert mevalonate into isoprene pyrophosphate. The important thing is to recognize that isoprene pyrophosphate is an intermediate. The next step, again, multiple steps, is going to be taking 6 isoprene pyrophosphate units and condensing them into a 30-carbon molecule. This 30-carbon molecule is called squalene. Squalene is a long, linear molecule that is 30 carbons long, that is synthesized by taking 6 isoprene units and putting them in, hooking them up, linking them in tandem. Now the next step also includes many steps to give us our final structure of cholesterol. Now recall, the structure of cholesterol is a multi-ring structure. So multiple ring structures from a linear molecule means that there's going to be a series of enzymes called cyclases that will be involved. And the job of the cyclases is to take the linear molecule and combine carbons to form the 4 ring structures as familiar to us of cholesterol. Notice also on the number 3 carbon of the cholesterol A ring, rings are labeled A, B, C, and D, the number 3 carbon on the A ring is a hydroxyl group. The enzymes that perform these reactions to add hydroxyl groups are called hydroxylases. So there are many steps involved, going from our mevalonate to final structure cholesterol. The most important elements that I'd like you to keep in mind are mevalonate to form isoprene pyrophosphate, which are the fundamental building blocks, building units of cholesterol. And then we are going to take 6 of these, link them together, end on end, to form our 30-carbon squalene, and then through many step reactions, we'll take this, convert it into the cyclized form of the rings to give us our 4-ring structure of cholesterol. When we add a hydroxyl group, we have enzymes called hydroxylases. This particular enzyme here is called 3-alpha hydroxylase.